Hi, everybody. This is Cynthia Cleto from Mount Sinai Innovation Partners, and I'm here with Dr. Hina Chaudhry for our Breaking Boundaries session, where we learn about innovation from highly active and diverse inventors within Mount Sinai. Dr. Chaudhry is the Director of Cardiovascular Regenerative Medicine at Mount Sinai. Hi, Hina. How are you doing?
Hi, Cynthia. Fine. Thank you very much for inviting me to do this.
My pleasure. We're so excited to get to know you, and I'm sure it's the same for the rest of Mount Sinai. We'd love to hear about what you've done in the past. So, we've been very curious in following your career and your research and wanted to know just a little bit more about you. Tell us about your background. How did you know that you wanted a career in cardiac medicine? That's very specific, Hina.
It is, I guess, being of South Asian descent and with cardiac disease running rampant in this population, it provides you with daily motivation. I myself was 7 years old when my late father experienced a heart attack. He collapsed on our driveway. He was having an inferior wall heart MI, which at the age of 7, it was a rather traumatic event because I had to basically grow up with the fact that he had heart disease and that we had to be quiet in the house and not, not annoy him or disobey him as children. He underwent quadruple bypass surgery when I was 8 years old, and it was pretty early in the days of bypass surgery. So there was still a very high mortality rate from the heart-lung bypass machine. It wasn't as routine as it is these days. And so I think that experience left quite a bit it was a turning point, let's say, in my own life, and I really wanted to explore how we could fix these problems. And thankfully, he had a small MI and he had extensive coronary disease, and he lived after that bypass. And then he underwent a second bypass when I was in my 20s. I mean, he took very good care of his grafts. He could have been like a poster child for how you protect your heart. So it wasn't until I was in my 20s, in fact, I was in medical school when he underwent a redo bypass because his grafts had occluded again. So as I said, I have lived with this disease my entire life, and since I was a small child, and that really, I think, would be the driving force. When I started my fellowship, there was kind of a roundabout circular path to my career. I was at Harvard for medical school. I had planned to never leave Boston for the rest of my life. I was in the HST program, which is the Health Science and Technology program, is geared towards training people for academic medicine and research careers. And but I ended up going to Philadelphia because my family was there. I had grown up there, and they offered me a chance at the University of Pennsylvania under the leadership of Dr. Judy Swain to start my fellowship a year early. I didn't do the traditional 3-year medicine residency, so I was in something called the clinical investigator track, which allows you to shorten internal medicine residency and start cardiology or any other specialized fellowship training a year early. Now, I'm not sure that still exists, so I don't want to get people thinking, oh, I could do this too, because I'm not even sure it exists right now, to be honest with you. But at the time, it did exist. I was fortunate that I was able to work with some really great mentors during my medical school years and get out first author publications as early as age 22.
Wow.
In the field of interventional cardiology. So I was really sort of headed towards interventional cardiology. And then with Judith Swain as my mentor at Penn, who had been an interventional cardiologist herself, but who has then switched to basic science after she did a sabbatical year in Phil Leader's lab at Harvard. She convinced me to become a basic scientist as well, because she felt that that was really where innovation lies. At that time, I was a fellow and was really excited about being in the cath lab. But I knew that in order to really be an effective basic scientist who could really translate basic science discoveries to clinical treatments, I wouldn't have the time necessarily to also excel in the cath lab. And I'm the type of person, I need to excel at what I'm doing. If I'm not going to do it at an excellent level and be outstanding, I don't want to do it. I don't want to try to do, I knew I wouldn't want to be doing both interventional cardiology and trying to excel at that and then trying to be a kind of scientist who was breaking grounds in important discoveries in the lab. I just didn't know of anybody who had been able to do that. And it's hard enough to be both a physician and a scientist. So then I explored. I really went after some very rigorous training in basic science. Judy Swain left Penn to become chairman of medicine at Stanford. I needed to stay on the East Coast because all my family is here. And then I trained in genetics, developmental biology, cell cycle biology with Deborah Wolfmuth at Columbia. So I did a very long fellowship in the genetics department at Columbia University while I was also junior faculty and a junior attending in cardiovascular medicine. And that's not easy to do. I mean, there are places that support it, like Columbia, to some extent, but it's quite challenging to actually do that well, even if you're doing something less, I would say, aggressive than interventional cardiology. And I chose to do imaging, and I love it. I really enjoyed the clinical interactions with my colleagues and maintaining my foot in clinical medicine through the echocardiography lab. What it does is it frees you up because I don't see patients in clinic, which I do miss, but I would not be able to be competitive at basic science if I did that. So my time in echo is such that it's contained. When I'm in the echo lab, I'm there. And when I'm out of the echo lab, I'm in my basic science lab. So that's sort of how I've attempted to combine being a physician and a scientist and I think it gives me the unique ability to translate discoveries in the basic science lab, and that's so important. I cannot overstress the importance. The one problem that a lot of hospitals are having in academic centers is they're really kind of funneling people into one thing or another. They want the clinicians to just do clinical work and bring in a lot of clinical revenue through RVUs, and then they want the basic scientists to just be in the lab and be generating NIH funds and other research funding. So there are very few physician scientists left. There's actually only 1.5% of MDs in the country who are physician scientists, and this is a huge problem because it's the physician scientists that are able to really translate discoveries at the bench to actually making clinical treatments. So we really have to focus on helping them and nurturing them and trying to increase their numbers.
Great. Thanks, Hina. That's such an interesting story, an interesting, impactful story about from your childhood and then the adventurous twists and turns that you found yourself in, kind of trying to navigate to what you really want to do, and how you want to impact the world. So I commend you for that. That's amazing.
Thank you.
One very curious question I had was, you did all this, and I'm also curious about your experience as a clinician, researcher, but also as a woman of color. How did all of this kind of unfold through that perspective?
I won't sugarcoat anything. It's not been easy. I would be a lot farther in my career had I been a white man. Let me just say that up front. There is no doubting that. I think when you're a woman of color in these stressful environments, and all my environments have been, challenging to say the least, I think you have to have nerves of steel because you're going to see people who are less qualified than you with less achievements get promoted faster than you. There is no doubt about that. And they are given leadership positions that are not available to me, and I'm fully cognizant of that. So I just focus on what I can control, and that is to really excel at what I do. So that people have to realize if they're in any way coming from a disadvantaged sort of, existence in academia, which I never imagined starting out at MIT with 2 degrees from MIT, then an honors MD from Harvard. I had never imagined I would go and face the kind of obstacles I did just for my gender, ethnicity, and background. I can't even imagine. Like at that time, I was just this idealistic person, just wanting to, make an impact on the treatments that are available in this disease. And I just never thought things could get so politically messed up in so many ways, just based on my background and my gender. So that's what I'm saying. I'm not going to sugarcoat it, but I think at the end of the day, you're responsible for your own career and you're responsible for rising above these obstacles. And that's something I counsel people who are being mentored by me, who are predominantly women of color, is that if you excel at what you do, there's nobody that can stop you. Not a single human being, no matter who they think they are not going to be able to stop you. You just have to really excel.
Great. Those are fighting words, Hina. Thanks for inspiring the folks out there. So in terms of your research, what are you working on right now, and what are the findings that you're the most excited about?
Well, there's so much that I'm so excited about, but let me start with what I consider to be really my baby, is what I started my postdoctoral fellowship training in under the mentorship of Dr. Deborah Wolgamuth at Columbia. So I learned everything I could possibly learn from her about cell cycle biology. Mind you, she was not doing any research in cardiovascular medicine or cardiovascular disease. Her background is in reproductive medicine and cancer genetics. And so she studied cell cycle as applied to those disease processes. And I was fascinated by cell cycle biology, so I asked to be, taken under her wing and do a genetics fellowship in her department. And she has been a mentor to me to this day. And I was able to sort of incorporate or bring in cell cycle biology into the cardiovascular field. There had been very little that had been published on it before. And so I did my earlier studies were on testing the effect of cyclin A transgenesis in mouse models and how we could continue to keep cardiomyocytes proliferating. So as you're aware, Mount Sinai owns intellectual property related to that because we've extended it now into humans. When after I came to Mount Sinai, I had performed the first porcine studies involving gene therapy using any cell cycle regulator. And in 2014, we published a paper in Science Translational Medicine, which even now in 2021, no other laboratory I feel has shown an effect that's significant in that we were able to use cyclin A2 gene therapy and we had 55% increase in new cardiomyocytes in the porcine infarct zone. And these were large infarcts that we were inducing, and yet we counted on a molecular level the number of cells produced, and it was a 55% increase. And the pigs survived. They were not, they didn't have any adverse effects from the gene therapy. We've done follow-up studies on off-target organs and saw that there were no effects. Those studies to examine any adverse effects are not published. Those are part of a package that we are, submitting to the FDA to show the safety of this as we try to, obtain investigational new drug approval. There's a lot that goes into that. Just because you publish efficacy in a large animal, you're not done. There's a lot that goes on after that in terms of showing safety. We had to change the vector. We wanted to use the latest generation vectors. And so that I'm really excited about because I think that's really close to entering human testing. We've tested the new vector in human cardiomyocytes that we cultured, and these are primary human cardiomyocytes that we obtained from patients who had died of non-cardiac causes, and it shows beautiful cytokinesis captured by live imaging. So I'm excited about that. I'm excited about a stem cell population that also Mount Sinai Innovation Partners owns the patent rights to, or the CDX2 cells we're isolating from human placentas now. We had shown in mouse models that these cells are very similar to embryonic stem cells. They retain all the stem proteins of embryonic stem cells, and they are capable of traversing through the circulatory system because they're responding to homing signals that are emitted by the injured organ. In our case, we've studied the heart, but now I'm excited about collaborating with other scientists to study other types of organ repair other than the heart.
So they're highly targeted to tissue
Right.
that is suffering, basically. Yeah, that's
Right.
great.
And we know what proteins are involved in these homing signals. We've done some pretty extensive proteomics testing as well. And so we know that these cells are highly cardiogenic. And now that we're isolating the human cells in a collaboration with our Department of OBGYN, both Dr. Rhoda Sperling and Dr. Sherry Gelber are involved in these studies, and that they're providing us with the patients and the placentas. And the human CDX2 cells we're collecting from human term placentas are forming beating organoids within 4 days. In fact, I think it was just the last 2 weeks, I sent the newest data to Mount Sinai IP saying, look, this is human, this is not mouse anymore.
We're at the next level already.
Yeah, so we're tremendously excited about that. I just really think hopefully in the next couple of years we should be able to use these different technologies to make a difference in at least heart disease, and I'm starting to have hope of other disorders. We're also looking into seeing what these cells we believe are highly neurogenic as well as cardiogenic, so we're exploring that as well.
So there could be various applications in other organs. That's really interesting. Great. How do you see this evolving, and what is next?
I think it's really, as an MD, I mean, I want to make sure that this is able to help patients. Because the only reason I started out studying basic science was not to sort of, just study a gene pathway just for, just to understand a gene pathway better, although it's fascinating. It was really to develop novel therapies. So until they become novel therapies, I don't think I can rest. So once they're entering that pathway to actually be able to save human lives, we're going to keep going.
That's great. So last question, Hina, what would be your advice to folks that are wanting to go through the same path as an innovator within the system and are also, minorities or underrepresented minorities?
Come talk to me. I think that, the sad thing is that I'm not counted as an underrepresented minority because I don't fall into those categories. So I don't benefit from those types of efforts. But I think that, there's always obstacles. There's always, people who make it more challenging. There's always people who make it easier. There's always people amongst the leadership you can go to and you can trust and get the kind of support you need. I think that's always found in every institution. So
thank you, Hina. It's been great kind of learning about you and your research. And thank you so much for your passion and drive and your contributions to healthcare. We're really looking forward to the outcomes.
Thank you.
That'll be great. Yeah. Thank you, everybody.
We hope to be publishing these results soon with the human CDX2 cells. So that's sort of like the most recent exciting thing that we've got coming out of the lab.
So once you have that, I think we should, we'll definitely, make sure that the whole of Sinai through our newsletters at MSIP know what the newest is in development with that. Okay.
Thank you. I appreciate it. Thank you very much.
And thanks to everybody who joined us. Have a great day.